Hello everybody, welcome to Knowledge Without Bar. Thank you for joining my tutorial. Today we will discuss about bioreactors under industrial microbiology. So let's start the session. First of all, we have to know what is bioreactor. Now what does the bioreactor means? Bio means any biological organism and reactor means a vessel where a reaction takes place. So bioreactor is a vessel used for the production of certain desired compounds using biological organisms. For example, bacteria, algae or any other microorganisms by providing them optimum conditions for the controlled and optimum growth of those microorganisms with regulating agitation, temperature, aeration and pH. Principally, a fermentation reaction is taking place within a bioreactor. Hence, many bioreactors are also called fermenters. So, usually fermenters and bioreactors, these two terms are used as synonyms, but there is a fine line of difference. Bioreactor means where a biological organism produces a certain chemical compounds by any biochemical reaction, mostly the secondary metabolism. But if those biochemical reaction is specifically fermentation reaction, then those bioreactors are called fermenters. So in that sense, all fermenters are bioreactors, but all bioreactors are not essentially fermenters because there are many other biochemical reactions besides fermentation. And recently, besides microorganism, different cell cultures like the animal cell culture, plant cell culture, etc., the independent cell culture are also utilized for the production of varied compounds within the specialized bioreactor. Hence, nowadays is also termed as microbial cell factories. So, this is the modern term of present day bioreactors. So bioreactors is a vessel that I already told you, and within this vessel the environment is controlled for the growth of specific microorganism or specific cells. So everything is controlled within this vessel, and definitely the vessels are infection proof. There must not be any contamination. So then these vessels are filled with sterile growth media and a specific microbes used as inoculum, or maybe different cell cultures, specific cell lines may also be utilized as a inoculum. And then this controlled environment means the environment is typically set for the optimum growth of the specific microorganism that we used for the product development. So by getting the optimal environmental condition, these microbes or cell lines are increased in number, so biomass increases and the specific biochemical reaction takes place. And in this case, the ethanol alcohol is our target compound. So there is another step, that is recovery of the desired component. So after the reaction has over, then these target compounds are recovered from the bioreactors, and then industrial scale production taking place, packaging takes place, and it's sent to the market. So this is the general phenomenon or flow of events that takes place within a bioreaction. So then a brief history and discovery of the bioreactors. The need of large-scale production of the different beneficial compounds or products were first observed during the World War I. Charles Weizmann, who is a professor of University of Manchester, first got the idea of bioreactors. He first produces the synthetic acetone by using the bacterium Clostridium acetobutalicum. the large amount is not comparable with the present day industrial scale, because in those days the large amount doesn't means in a billion or million liters. Like a 20-liter capacity fermenter was developed by Devekes and Liebman during 1944. So in those days, another obstacle was that the introduction of the sterility. That was the major constraint in those days. In 1950, the first pilot fermenter was introduced by Hindustan Antibiotics Limited, situated in Pimpri, Pune, India. So this is the very brief history and of discovery of the bioreactors. So now the important thing is the design of the bioreactor. Bioreactors may be of different types, and in present day, as the science has become advanced, so different kinds of bioreactors have been introduced. But there is some basic principles or characteristic feature to design a effective bioreactor. What are those? First of all, the bioreactors must be smooth surfaced, non-toxic in nature, corrosion proof, and easy to sterilize. That means it must be made up of glass or stainless steel. Second is optimum cell biomass attained to providing the optimal and controlled condition. So the vessel must be designed in such a way that within the vessel we can capable of maintaining the optimum cell conditions or biological conditions. The microbes can grow fast within those vessels. That's important. Provision of regulating pH and temperature, mixing and aeration. Flexibility in changing the metabolic condition with time. High energy efficiency, that means low power consumption. A proper inlet and a proper outlet. Inlet is required for introduction of growth media, required for the cell cultures or microbial cultures, introduction of the inoculum, and outlet is mainly meant for the recovery of the products. And final point is minimum use of labor in operation, harvesting, cleaning and maintenance. So these are the basic characteristic features in designing any bioreactor. Now there may be a question, how big a bioreactor may be. So they may be 1 to 2 liters to 5 lakh liters or even more, 1.2 million liters. How amazing it is. So 1 to 2 liters bioreactors are generally laboratory scale bioreactors where the testing and standardization is taking place, and stepwise we scale up to produce industrial scale bioreactors where a much more capacity has been introduced. Recently, a conceptual air-lived bioreactor has been designed to produce animal cell culture, that is artificial meat production, which has the capacity of 15 lakh liter. Can you imagine 15 lakh liter? And recently the production was scaled down to 3 lakh liter because this is under the standardization process by utilizing the computational fluid dynamics model. So it has been reported by Lee et al. In 2020. So if you want to check the article, you can check by using this link. Now, what is the structure of a standard bioreactor and how a standard bioreactor looks like? So this is a picture of a standard bioreactor. So as I already told, there must be a vessel, and that vessel is usually made up of a glass or steel, but normally industrial scale bioreactors are made up of steel because glass is not very much user friendly, because there is every chance of break in downstream processes. So in the industrial scale, usually the bioreactors are made up of steel. So the steel vessels are covered with a specific jacket, which is called the outer jacket. This jacket is there to maintain the temperature within the vessel, and in the jacket there is a water inlet and a water outlet. So cool water enters within the jacket. It covers up the entire vessel where temperature is required to be reduced, and after the dissipation of the heat, the water is out through the water outlet. And there is specific another inlet for introduction of the media and introduction of the inoculum, and the outlet from where mainly the recovery of the desired compounds are taking place. And this inlet and outlet are definitely covered with valves and filters to reduce the contamination. And there is several other sensors for maintaining the pH. There may be a pressure indicator. Is mainly used. Pressure indicators are mainly used where when the reaction taking place within the vessel produces gases. So those gases are constantly accumulating within the vessel and creating the pressure. So there may be a pressure gauge to release the extra pressure through exhaust line. And if the metabolic reaction taking place within the vessel increases or decreases the pH, there is an auto titration unit to maintain the specific pH or neutralize the media for the active growth of the bacteria. And there are another important thing, look at this axis fixed with a motor, and this axis are having many blades. So these are called the impellers or agitators. So what does these impellers or agitators do? When the motor is circulating, these agitators are also circulating. So with the circulation of the agitator, the media present within this vessel are also circulating, and this circulation is absolutely needed for the optimum growth of the bacteria. Why? Because if there is no agitation within the media, then bacteria will grow in discrete fashion. So agitation leads to the uniform distribution of the growing bacterial population within the vessel. So that's why agitation is needed. You all know when we put a liquid culture for our laboratory use, we always place those conical flasks in a BOD incubator shaker. So shaking is necessary for the uniform dispersal of the growing bacterial cell. So along with these impellers, there are two additional rotating units which are called the baffles. And why these baffles are used along with the spargers we will discuss shortly. So in the base of the bioreactor there is another specialized structure which are called the sparger. So sparger are usually used where the aerobic culturing condition is needed, because in the anaerobic condition there is no need of the sparger. So sparger is nothing but from where the air bubbles are circulating within the vessel, because with the growing population of the bacterial cell there is a need of constant need of oxygen for the respiration of those growing population of the bacteria. So the air bubbles are specifically oxygen is purged within the vessel with this device, and this air are definitely sterile air, because non-sterile air will going to contaminate the media. So there may be a different type of spurger, like porous spurger. Porous spurger means a flat surface with multiple pores, or from where the sterile bubbles are coming out and circulating within the vessel, media within the vessel. There may be orifice spurger. Orifice spurger means instead of this flat base, there is a pipe containing several pores, and this pipe containing several pores may be stagnant or it may be rotating also, and from those pores, the air bubbles are coming and mixing within the media in the vessel. Or simply there may be a nozzle spurger. Nozzle spurger means there is a single nozzle from where the sterile air are mixed within the vessel. So these principally three types of spurgers are used, but there are several different kinds of impellers are there. We'll shortly discussing in this tutorial. And so these are the specific structural requirements for a bioreactors that I already discussed, like impellers, baffles, spurgers, sensors. Within the sensor there may be spacer sensor, pH sensor attached with the auto titration unit, temperature sensor, airflow sensor, agitation rate, dissolved oxygen rate sensor, and foaming sensors. So foaming is also a consequence of many metabolic reaction. So there is a foaming sensor to reduce the chances of forming the foam. And number 5 is the most important thing, is the filters. As I already told, that in the inlet and the outlet they are fitted with some filters. Along with the spurger line, there is a filter attached to reduce the contamination. So there may be 3 types of filters are mainly used: the membrane filter, package belt filter, and cartridge filter. The membrane filter is cellulose acetate or nitrate made. The pore size is 0.2 mm. Is utilized particularly to remove the particulate matters. The package based filter are a glass wool or cotton wool. The pore size is not uniform, as it is a glass wool or cotton wool. So, and is not utilized recently in bioreactors or high-end bioreactors, because it's susceptible to compaction and channeling due to weight problem, because there is every chance to leakage of the media, and that media may comes out through the pipeline and comes in contact with this glass wool or cotton wool, and this glass wool or cotton wool will maybe get wet. And for that problem, in present day, these glass wool or cotton wool filters are not typically used. Instead, they used cartridge filter made up of polycarbonate materials, and is available in varied pore size. And depending on our requirement or specific requirement, the pore size can be determined, and they are more durable and reliable, but it's a little bit expensive. So now come into the type of impellers or agitators. There are mainly 4 different kinds of impellers or agitators. The first one is Ruston blade or disc turbine. So we are talking about these structures. Okay, so the disc turbine are nothing but 2 blades are protruding outwards from a axis. So this is the side view and this is the top view. If we look from the side, so this may be the side view, and if you look from the top, this is the top view. So this is the side view of this turbine, and this is the top view of the disc turbine. The number of blades may vary, like 4 to 8 to 6. There are different number of blades may be there. So when this disk turbine impeller rotates, so they creates a torque towards the outside or towards the periphery. So that is the principle of disk turbine. Second is the varied disk turbine, where the blades are inwardly located. This is the side view. If we look from the top, the blades are inwardly located. Like in disk turbine, they are outwardly located, and in varied disk turbine, they are inwardly located. So when the varied disk turbine rotates, so it creates a torque or a rotational wave center towards the center. So that is the basic difference between Raston blade disk turbine and varied disk turbine. So it creates a torque towards the center, and it creates a torque outwardly towards the periphery. Number 3 is open turbine with variable pitch. So it's very much similar to the disk turbine, but in that case the axial part is coupled with a flattened blade. So this structural alteration is capable to cause a very effective circular motion. So open turbine variable pitch impeller are very much effective in producing the uniform torque within the bioreactor. And this is a special type, is called the marine propeller. It moves like this. So this also creates a torque towards the periphery, but the uniformity is somewhat higher in case of the open turbine. Now the question arises that why these axial and radial impellers, like why these axial and radial impellers, these are axial and these are towards the radius or radial impellers, produces vortex. So when this impellers rotates, it always creates a circular movement or circular wave, and this circular wave destabilizes the media present within the bioreactor, and which causes a upward spillage due to the huge vortexing effect. So when the axial and radial impellers rotates, it creates a, you can see when it rotates, it creates a vortex within this media, and due to the continuous and rapid vortexing, it creates a outward spillage, which is not desirable in a bioreactor, because spillage in one hand may cause the chances of contamination, in another hand it may retard the growth of the microorganism or cells, because there is every chance of damage of the cell structure of the microorganism. So in that reason, the baffles are also used along with the impellers. So what happens when baffles are there? So when the impellers rotates, it creates a vortex, and at the same time the baffles are also rotates. So impellers, if you look at carefully, impellers are circulating, and those creating a circular movement of the media present within the vessel, and this movement is axial towards the axis, and when the baffle circulates, it creates a horizontal wave. So the impeller, movement of the impeller causes a axial vortexing that I already told, and the movement of the baffles causes horizontal wave. So this horizontal wave neutralizes the axial wave. For that reason, there is a very minimum chance of spillage and vortexing effect. So for that reason, the impellers are always coupled with baffles for the uniform growth and optimum growth of the microorganism and increase the efficiency of the bioreactors. So for today, thank you for listening, and in next session we'll discuss about different types of the bioreactors and some other aspects of the bioreactions. Thank you for listening.